This is podcast number 28. In this podcast, we'll talk about membranes that surround cells, how one of the properties of membranes is how fluid they are, how much they can move in a couple dimensions. That's important for how the membrane works, and how the membrane works is important for how the cell works. And cells can adjust their membrane fluidity by adjusting their chemical composition. Sorry, I didn't write that. Adjusting their chemical composition. Temperature also adjusts membrane fluidity. Okay, one of the techniques that cell biologists use is to label different parts of the cell with antibodies that have colors on them, fluorescent colors, so that you can see different parts of the cell well. And in this picture of cells growing on a dish, microtubules made out of the protein tubulin are shown in this green fluorescent. The DNA is shown in blue fluorescent, and actin fibers, which are just underneath the surface of the membrane, and also you can see these little red lines here, are throughout the membrane. But it's trying to show you how these cells all don't have the typical round shape that you would typically see on a diagram of an animal cell, for example. Cells can have all kinds of different shapes. You have cells in your body that are, depending on how tall you are a meter long, that they start off in this lower part of your back and go all the way to your big toe. So cells have all kinds of different shapes depending on what they do. And some cells have to transport a lot of material across their membranes. Here's an example of a cell that lines your gut, and it has a very wavy membrane. And what that very wavy membrane does is it provides a lot of surface in order to, and that surface is used to transport things across your gut, like things that you would eat, into cells. You get a lot more surface by making it wavy than if it was just flat. Now, what they're talking about here is the volume of the cell. So the volume contained inside this little finger projection doesn't change much, but the surface here, as indicated by this outline of this membrane, increases a lot. Part of what gives a cell shape is what's called the cytoskeleton. Just like you have a skeleton that gives your body shape, cells have a skeleton that skeleton is dynamic and it can move, and microtubules and actin filaments are part of that skeleton. Okay. Most of this podcast is going to be talking about the phospholipid bilayers. Again, this is a phospholipid has a water-loving phosphate-covered head, and then the fatty acid tails are hydrophobic. They come together and form the two halves of a membrane. The part in the middle of that is lipid-like. Inside eukaryotic cells, there's a membrane structure outside the nucleus called the ER, or endoplasmic reticulum. And that's a site that makes a lot of protein, stores calcium, and it's typically lots of wavy membranes. So there was a group of scientists that did a study looking at how the endoplasmic reticulum might have formed, and how is it that it gets this wavy sort of shape. And they made sort of a fake endoplasmic reticulum on a slide, where they took just vesicles and a bunch of microtubules, and then they had the microtubules covered by the vesicles, and then they stretched out the vesicles using proteins in the cell, and I'll show you how they did that. Here is our phospholipid bilayer. Here's the phosphate head group. Here are the fatty acid tails. 2 layers of there, that's why it's called a bilayer. This is like a typical membrane, and the endoplasmic reticulum inside the cell is a membrane, just like the membrane that surrounds the nucleus, or the membrane that surrounds the mitochondria or the membrane that surrounds the chloroplast. And then this would be water, and this out here would be water inside the cell. There are motor proteins that can move things in the cell. This one is called kinesin. There's two major motor proteins in cells. One's called kinesin, the other one's called dynein, and they have specificity for how they move on microtubules. This is also a protein-based structure inside the cell. Remember, microtubules are made out of tubulin, and they pull chromosomes. They also form the part of the cytoskeleton of your cell. And kinesin molecules can walk on microtubules. Microtubules have a plus end and a minus end, and kinesin always walks toward the plus end, and dynein, the other molecule, walks toward the minus end. And in real live cells, kinesin and dynein are used to move things around inside the cell. One of the ways things get moved around in the cell. In this setup, they use those motor molecules to pull the membrane, sort of stretch out the membrane like you're stretching, like you had a balloon and you're stretching out part of that balloon and deforming it, and they're trying to see what kind of shapes they could get out of that. Okay? Using these molecular motors. And the motors work on the track, which are the microtubules, and they pull the membrane with them. So I'm going to show you a movie which is an animation of this kinesin molecule moving. This is a really famous one. You might see it in a bunch of cell classes because it's so well done. But this is kind of freaky looking. You're going to think this isn't real, but it actually is a good approximation of how things look inside a cell. So what you're going to look at is, here's the microtubule, the track that this thing is walking on. This thing here is the kinesin molecule, the motor molecule, and the thing it's attached to is a vesicle, a big blob of membrane. So you can watch this move here, pulling the vesicle along. So we have really good data that that's actually how these proteins move. Okay, and it's walking along that microtubule by splitting an energy molecule called ATP that causes the thing to change shape. And as it changes shape, it actually walks down the microtubule. Okay, let's stop that and go back to our PowerPoint here. All right, in this experiment where they had microtubules and vesicles, and they're trying to stretch out those vesicles, they're trying to see can changing the shape of a membrane get to something that looks like the ER. So they're taking video of the microscope, and they're looking at a particular spot over time. And if you look at this little spot here, it's pretty small at time 0, and 43 seconds later, it's grown to this length. So this is by kinesin pulling that vesicle along and stretching it out. Okay, here's another one. If you look at time 159 versus 246, you can see the membrane is getting pulled in these directions. And you guys haven't looked at endoplasmic reticulum, but this could be like endoplasmic reticulum in the cell is a bunch of flattened tubes inside the cell on which ribosomes sit and make proteins. They can also do this with a different setup. This is a fluorescent bead that has a membrane attached to it, and it's you can move this fluorescent bead with the laser. The laser actually exerts a force on the bead, and you can get the bead to pull away from a vesicle, and you get a tube formed as a result of pulling that away. So think of this as a balloon, and your fingers are pinching a little piece of the balloon and pulling that away. If they change the membrane composition of the vesicle it becomes more relaxed in shape and they get a different size tube. So they're showing here by changing the membrane composition of the vesicle from one kind of phospholipid to another kind of phospholipid, they get different size tubes. And then they can actually measure the force that it takes to pull out those size tubes by measuring how much light pressure using the laser do I have to exert in order to draw out a tube. And that's measured in force on the y-axis, and then how long does it take to draw out the tube on the x-axis. So they start pulling out those beads there, and from a relaxed vesicle, to pull out a tube doesn't take much force at all. If it's a so-called normal vesicle, different kind of membrane composition, tougher than the relaxed vesicle, it takes more force. And then if you change the membrane composition again, it takes a lot of force. So how do they change this membrane composition? Well, you can change the actual kind of phospholipid that's in the membrane. That'll do some of it. But a big way that cells change their membrane composition is with cholesterol. So you have thought probably mostly as cholesterol is a bad thing, but you need cholesterol in order to make membranes. It's one of the things that regulates how stiff that membrane is. If it's loosey-goosey like this, if it's so-called normal like this, or if it's very, very stiff like that. And you can, cells can adjust how fluid their membranes are, how easy it is to deform those membranes by adjusting how much cholesterol is in there. Here is a table of a whole bunch of different kinds of phospholipids, and then here's cholesterol and the effect that it has on their membranes. Different types of membranes have different chemical composition. So even within one species, the endoplasmic reticulum of a rat has different chemical composition than the cell membrane of a rat. Or the Golgi apparatus, which is another membrane-like organelle in a cell. Just to give you an idea of how much membranes change over time. Membranes are not rigid over time, they change. I'll show you this little movie. So here membranes are labeled in fluorescent green, looking at a cell, and over time you can see they are very dynamic, that they change pretty dramatically over time. Okay? So you can get a false impression of what things look like, a false impression in your head when you're looking at a rigid diagram. In real life, cells are much more like a lava lamp, that things are constantly blebbing off of each other and fusing together and changing all the time. Okay? The composition of these membranes has been subjected to natural selection because these membranes have to do different things. The membrane of a mitochondria has to do something different than the membrane of the endoplasmic reticulum or the membrane of the cell membrane. They have different jobs to do, and those jobs require a different amount of fluidity in the membrane, and therefore the membrane composition is different in those different membranes to arrive at a membrane fluidity that will work for those membranes. Okay, another example of how lipid composition can affect membranes is in this experiment where they took a bunch of vesicles and they had fluorescent blue for lipids that tend to be more rigid, and fluorescent red that tend to be for lipids that tend to be more relaxed. And they put them together in different combinations to give different shapes just by changing the lipids. So you can get this shape, you can get these shapes. Notice it said warm to 37 degrees and it'll divide in two. So temperature also affects the shape of these membranes. Okay, they can assemble into all these different wild shapes, like even things like this. And these look like cells. These look like the cells that we see out in nature. So just by changing the composition of membranes, you can get things that we recognize as similar to the shapes of cells. There are algal cells that look like this, for example. And this is just by changing the composition of membranes. So if we've been talking about membranes as just a bilayer, just phospholipids with fatty acid tails, and the middle is hydrophobic, and the outside likes water. But membranes are actually lots more complicated than that. So here's a couple cholesterol molecules stuck in that membrane. Cholesterol molecules can also adjust how fluid this membrane is. And by fluid, I mean that there are lots of proteins stuck in a real live cell membrane. It's not just lipid. In fact, for some membranes, most of what the membrane is by weight is protein, not lipid. So even though these little squiggly balls with the threads hanging off of them are all lipid by weight, if you weighed this whole membrane, most of what is there, or at least half of what is there, would be protein. So the proteins, in order to do their job, they are going to move in the membrane. So they can move laterally across this plane, and they don't move so much up and down, but they do move a lot side to side. And they have to move and flex in order to do their jobs. And so membrane fluidity is a big deal. And you can change membrane fluidity pretty dramatically with changing what kind of phospholipids are in there. And it affects how they interact with these proteins that have to move in this membrane and how that membrane can do its job. So if we go back to the first couple slides here where we're looking at vesicles, or looking at microvilli in intestinal cells, that membrane there is packed full of protein. Those proteins are recognizing things that are in your gut that you want to get into the cell. They're not just going to fall into the cell, they've got to get into the cell through a protein that is stuck in that membrane. And if you want to get, say, lots of sugar from what you ate into the cell, or fats from what you ate into the cell, or proteins from what you ate into the cell, they've got to go through the membrane through a protein. And those proteins have got to be able to move in that membrane. They've got to be able to flex and change shape, and they've got to move from one place to another in order to do that job. So the job of the cell is to absorb things from the gut. They're going to absorb that through proteins, and the proteins have got to be able to move. Now, one way I have found useful to think about, rats, here we go, sorry, found useful about how proteins move is to think about a kid's ball pit. This is an awesome ball pit. This is easily one of the major reasons I had kids, was so that they would allow me into the ball pit to watch my kid. Because if a 56-year-old man goes into the ball pit by his own, they call the police. So, and my kids are too old for that now anyway. But if you think of a ball pit like a cell membrane, and you walking in the ball pit, you're a protein in that cell membrane. As you walk through the ball pit, the balls get out of your way as you walk through it, and they fill in all the spots in between as you walk away from a particular spot. So you are the proteins moving in the membrane. You do the job of letting stuff in or out of the cell through that protein, and you've got to be able to move through that membrane in order to do that. If all these balls were frozen in place, you could not move. So the balls have to be stuck together well enough so that it doesn't let the contents of the cell out, but not stuck together too much so that you can't move. And that's how the composition of these balls, the phospholipids, can affect how well they stick together, and also whether or not and how much cholesterol is in there, and also temperature is in there. So think back to the podcast where we talked about how cells detect fear, and you had that big signaling cascade that started with epinephrine on the outside of the cell, and then the epinephrine receptor, and then G protein, all that sort of stuff. If the epinephrine receptor can't change shape because it's stuck in a stiff membrane, none of that works. And if the G protein can't move across the membrane to interact with that receptor, none of that works. So membrane fluidity is a critical feature of how membranes work, and that fluidity is dictated by the composition, the chemical composition of that membrane.